Again, Mendel tried several combination. Then a dihybrid cross, let us see, with two pairs of contrasting characters. Here you can see one such combination was crossing a variety with a round and yellow seed with another variety having wrinkled and green seed. Here two characters are taken here. Round and wrinkled, that is a contrasting pair. Yellow and green, that is another. Color of the seed is character, that are different. Contrasting traits are yellow and green, and shape of the seed is one character and its contrasting are round and wrinkled. So here you can see how the experiment, it is a dihybrid cross that is done by Gregor Mendel. Here you can see round and yellow seed. Its picture is also given here. Round and yellow, that is a dominant trait. Two dominant traits are there, pure round. So that it is two genes are capital R, capital R. Pure yellow, it is shown by capital Y, capital Y. Then its contrasting pairs of character are round, wrinkled is the contrasting character, then yellow, its opposite character is green, that is represented by small letters. Here it is cross pollinated. The F1 generation was capital R pair with the small r. The gametes at the time of genetic, we know that during the meiosis haploid division occur and these pairs will get separated. So the capital R combined with the small r, capital R from the male and female, when it combine, a combination get capital R, small r and capital Y, small y from the female. The first generation, its genetic constitution is this one, capital R, small r, capital Y, small y. All round and yellow seed externally, but the green and wrinkled character is there inside the body. Genetically, they have these recessive traits also inside them, but externally all round and yellow seeds are produced. Then again, here the second generation by pollinating or self-pollination, here the next generation or F2 generation is produced here. You can see that the gametes formed here is round, maybe pair with this capital or this one. That is shown here, round with the capital R or round with the small y. This again, small r may combine with y or small r, small y also may be produced. That is shown here in this case. So four kinds of gametes may be produced: capital R, capital Y, round and yellow, round and green, then wrinkled and yellow and wrinkled and green. These are the female sex cell. The same kind is produced here, taken in the or coming in the male sex cell. The combination or by the self-pollinating this pea seed, we will get the result is like this: capital R, capital R, capital Y, capital Y. This is here in this seed. Then the seed will be round and yellow. Capital R, capital R, capital Y, small y. That is in this result. Then this combined with this one, capital R, small r, capital Y, capital Y. That is round and yellow seed. Then capital R, small r, capital Y, small y. This is also round and yellow seeds are here. capital R, and small y and capital Y, round and yellow. Again, capital R, small r, then small y, capital Y. Here, again round and yellow seed. Here, capital R, small r, then small y, small y. Here, you can see round and green seeds are produced. Thus, we can find out by seeing all these, we can count that 9 phenotypical ratio 9:3:1. That is 9 dominant rated seeds we can see 1, 2, 3, 4, 5, 6, 7, 8 and 9 yellow round seeds we can see. The dominant traits 9 seeds are there. Then if we count round and green seeds 1, 2, 3. 3 round and green seeds are there. Again yellow and wrinkled seeds 1, 2, 3. Yellow and wrinkled seeds we can see 3. Then only one is there with the recessive traits, pure recessive traits, only one seed is here. That is green and wrinkled. That is the ratio 9:3:3:1. That is the dihybrid ratio. If you ask to, you need not to, usually in the examination, you need not to fill the Punnett square of dihybrid ratio. You have to write only the monohybrid ratio. You have to write the Punnett square or draw it. But dihybrid, you need not to, you will not have for the examination. But phenotypic ratio, you have to write the dihybrid phenotypic ratio 9:3:3:1. You have to write the phenotypic ratio, dihybrid phenotypic ratio, that you should learn this one. Then again, we have to learn Mendel's law of inheritance. These are the 3 important laws you have to learn. It will be asked to law of dominance. You have to write the law of dominance or law of segregation. Then law of independent assortment. It will be asked to write the Mendel's law of dominance. Such questions may be there for the examination. Law of dominance state that out of a pair of contrasting characters present together, only one is able to express itself while the other remain suppressed. The one that expresses is the dominant character and one unexpressed is recessive. The recessive character can express only when a pair of consists of both recessive. When the pair consists of both the recessive. The out of a pair of the letters in italics is the main part of the law. You must write this part for the examination. If you are expressing or writing the law up to here, it will be better. When you write it for the examination, you have to write this much. From here to here, you have to. Out of a pair of contrasting characters present together, only one is able to express itself while the other remains suppressed. The one that expresses is the dominant and the one unexpressed is recessive trait. Then the second law is law of segregation, also called law of purity of gametes. That is the second law. The two members of a pair of factors separate during the formation of gametes. They do not blend but segregate or separate into two different gametes. This much is needed. For the second law, you have to write this much. Two members of a pair of factors separate during the formation of gamete. Gametes, at the time of gamete formation, the factors meaning two genes that separate. At the time of gamete formation, they do not blend. Their character will not get blend but segregate or separate into different gamete. Then the third law of independent assortment. When there are two pairs of character, the distribution of a allele of one character into the gamete is independent of the distribution of the allele of the other character. Here you see that R may be combined with the capital R or R may be combined with the small y. R with the capital Y or capital R with the small y, or small r with the capital I or small r with the small y. So the distribution, it is saying that the distribution of the allele of one character into the gamete is independent of the distribution of the allele of the other pair. Then again there may be a question, what are the applications of Mendel's law? Why it is apply this knowledge, Mendel law? Then it is helping to predict the progeny. Then again it is used, this information is used by the plant breeders. Then new plants or new combination of useful character can be produced by hybridization. New varieties are produced. Then again another term you have to learn is the mutation. It is the sudden change in one or more gene or in the number of structure of chromosome. Many sudden change that happening in the structure or in the gene is the mutation. The mutation alter the hereditary material of an organism, then and result in change certain characters or traits. Then one examples are given here, sickle cell anemia. It's a blood disease caused by the gene mutation. That is change in the DNA resulting in the production of sickle shaped RBC. That is a disease. The RBC cannot take enough oxygen. Then again many other changes also occur due to the radioactive radiations. Also alter the gene structure, and their effects can be seen in generation after generation. An example, an atomic explosion which had occurred during the World War II had led to a number of deformities in the body of plants and animals which are still persisting. Because of the radiations, certain alter in the change in the structure of gene or certain other cases also, in some other cases also certain changes, sudden changes may occur. Such changes is known as sudden change is known as mutation. Now I am finishing the lesson, and for your examination, first time examination, you will have 3 chapters for your examination.